Alright, so now that we know some basic anatomy of the skeletal muscles, the question that we will be addressing in this segment is how do skeletal muscles generate forces when they contract? So the answer to this question lies in the structures called the sarcomeres. And the sarcomeres are what shortened to generate force. So, however, we need to understand first, what is a sarcomere? So recall that we had muscle cells, which we also refer to as muscle fibers, and those muscle fibers are packed with these rod-like organelles that are called myofibrils. Myofibrils are composed of thousands of smaller subunits that are linked end to end, and it's those small subunits that are the sarcomeres. A single sarcomere is found between two structures in the myofibril called the Z-discs. So here we have a more simplified diagram of a sarcomere. At the top, we see a sarcomere that is within a muscle that is relaxed, and at the bottom of the diagram is a sarcomere in a muscle that has contracted. So notice that the only difference between the two is how close the Z-discs are in the muscle that's contracted. The Z-discs are much further apart from one another in the relaxed muscle. So when muscles contract, the Z-discs of the sarcomere get closer together, and we're going to see how this is accomplished in just a moment. This process requires both ATP as well as calcium. If one of these substances is not present, the muscle will not contract. The sarcomeres are constructed from even smaller subunits that are called myofilaments, and there are three types of myofilaments that we will examine. The thin myofilaments, the thick myofilaments, and then finally the elastic myofilaments. So let's examine the thin myofilaments first. So the thin myofilaments are constructed from bead-like proteins that are called actin. The actin forms strands, and the thin filament consists of two strands of actin proteins that are twisted together. Also associated with the thin filament are these regulatory proteins that are called troponin and tropomyosin. And tropomyosin is a thread-like protein that is supported by the troponin protein. So these two regulatory proteins prevent the muscle from contracting when the muscle is not stimulated. And we'll also return to these a bit later to see exactly how they work. So the thin myofilaments are anchored to the Z-discs on either side of the sarcomere, and those thin myofilaments extend toward the center of the sarcomere. So next up we have the thick myofilaments, and the thick myofilaments are bundles that are made of a protein called myosin. A single myosin molecule looks like two golf clubs that have been twisted together at their handles. The region of the myosin that is twisted together with another myosin molecule is called the tail region. Myosin also has a flexible hinge-like region that is in close proximity to a club-like structure called the head of the myosin. The myosin heads have binding sites for actin, that is where myosin will actually grab onto the actin thin filaments and pull, which is going to cause the sarcomere to shorten. There's also an ATP binding site on the myosin head, because ATP is actually used to break the link between myosin and actin, so that the myosin can reset in preparation for another contraction. The thick myofilaments are suspended from the center of the sarcomere, with their head regions extending in many different directions toward the thin myofilaments that surround them. So now the third myofilament is referred to as the elastic myofilament. It's made of a large protein that's called titin, and the elastic myofilament does more than just suspend the thick myofilament from the center of the sarcomere. So notice that the elastic myofilament is anchored into the Z-disc and also attaches itself to the thick myofilaments that are suspended within the center of the sarcomere. However, as I just mentioned, it does a little bit more than this. So notice that the region of the elastic myofilament that's in close proximity to the Z-disc looks a lot like a spring. And the reason for that is because the elastic myofilament is the filament that gives the muscle its spring-like properties. So that little spring-like region actually does behave like a spring. So if you were to look at a light microscope image of an actual sarcomere in a real muscle, you would see two distinct bands on either side of the Z-discs. And here in the image that is on the lower part of this diagram, you can see the Z-discs as these very dark, thin vertical lines. The dark region that's in the center of the sarcomere is called the A band, and here I always think dark, since there is an A in dark. And the A band represents the region where the thick and the thin myofilaments overlap. The other region, which is the light-colored region that exists on either side of the Z-disc, is called the I band. And here I always think light, since there is an I in light. And the I band represents the region where only the thin filaments and the elastic filaments are found. So here there is no overlap with the thick myofilaments, which is why this region appears much lighter in color. So now that we have seen the components of the sarcomere, what actually happens when muscles contract? It turns out that the myosin of the thick filaments will attach itself to the actin of the thin filaments, and that little flexible hinge-like region that we saw in the myosin will pull. So since the thin filaments are attached to the Z-disc, the sarcomere will shorten when the myosin pulls on the actin filaments. So the term or the name for this phenomenon is called the sliding filament model, due to the fact that when the actin filaments are pulled, they will slide over the thick filaments during contraction. So notice that the thick and the thin filaments themselves did not shorten, they actually remain the same length. Only the distance between the two Z-discs in the sarcomere has shortened, and that is what generates a skeletal muscle contraction.